All right, welcome back. Part 2 of 2016 higher level paper 1. So on question 10 now, looking at the Krebs cycle. It's important to remember where this is happening. It's in the matrix. We are building molecules. We're building NADH up here. Oops, NADH. We're building FADH2. We're making ADP. And all of these things, well, not all of them, the NADH and the FADH2, those are all going to go on to the electron transport chain and provide the electrons, hydrogen ions to go into that intramembrane space to then come through that ATP synthase. So you can see starting with this 4-carbon molecule, we put in this 2-carbon acetyl-CoA, go through a whole bunch of oxidation reduction steps, and this is where we get into oxidation and reduction. If you are thinking about it, always think about oil rig. That's how I do it. Oxidation is loss of electrons, loss of hydrogens. Reduction is a gain in electrons or a gain in hydrogens. So we see here being reduced, this FADH2 is also reduction reactions. You're building something, and we're like adding the hydrogen ion right to that. So process number 1 over here, we're seeing reduction. We're creating a molecule, we're building it up. And over here, we're seeing a decarboxylation where the CO2, we're seeing the carbon 5 go to carbon 4. So that's what, see, a decarboxylated means you lost a carbon. Carbon 5, carbon 4, and that carbon came from that CO2 molecule. Oops, I'm sorry. Can't see everything. Trying out this new camera and everything. My new setup. So oxidation, loss of electrons, loss of hydrogens, reduction, gain of electrons, gain of hydrogen. And just also think about breaking things down. That's going to be an oxidation reaction. If you're building a molecule, that's going to be reduction. Number 11, the most bubbles. Which gas? Most of the bubbles, that would be carbon dioxide. And just think about yeast, regular respiration, making the CO2 and the water. Let me try to slow down a little bit on that. And yeast can also go through fermentation if you let them go long enough. Okay. So the next question on the Calvin cycle. So which molecule will first contain carbon 14? Now that's normal carbon. It's carbon 12. This would be an isotope that researchers might want to use in order to research. And the very first step of the Krebs cycle, you take this RUBP, add the CO2, and then you get the G3P. So that's why the answer is glycerate 3-phosphate. That would be the first one that would have this radioactive isotope here. And then it converts to PGA, then 3-TP or triose phosphate, and then you get the sugar, and then you have the regeneration of that RUBP. So Rubisco, that's an enzyme that works in the process. Glucose is what you get, not right away. You get, I think, you get a 3-carbon sugar, and then it combines quickly to make glucose. Now, if you see something like this, it looks like a cell, right? But it's not very sophisticated. There aren't a whole lot of, structures inside of there. So you might want to assume that that would be a prokaryote. I think I got that written down somewhere. I don't. But what we see here is binary fission of a prokaryotic cell. You have the DNA copying, but it just kind of squeezes two new cells. And that's a process called binary fission. So no mitosis. You don't have chromosomes moving anywhere in binary fission. And we might also see, they might put on this diagram, pili, or you see a flagella, and that would also tip you off. So it's a close-up image. So if this were a eukaryotic cell, we would be seeing something in there. Love these little cans. This is a regular sized can, and my hand grew. But so you wouldn't have telophase or mitosis. Remember cytokinesis? That's something we forget about, or not forget about, but when you have mitosis, remember in plant cells you have a new cell wall being built to separate the cytoplasm. That's really what you're doing, dividing cytoplasm. And in an animal like our cells, it kind of gets pinched because we don't have a rigid cell wall, so it can be pinched with no problems. So normally, we got cell theory. All things come from cells. Cells a basic unit of life, and cells can only come from other cells. Those are the basic ideas. So muscle fibers are a little bit different because they have many nuclei in one cell. So most of the time we think about the one nucleus, one cell. So there are exceptions, like to any theory. Here you go. Life is made of cells. Cells are the smallest unit of life. Cells can only from cells. I think I corrected it twice here. Cells can only come from other cells. I still didn't get it right. Cells come from cells. So basically saying scientists have not been able to create cells in the laboratory. Here we have a nice diagram, fluid mosaic model. We have our phosphates here. I think I got it here. Yeah, hydrophobic in the middle, hydrophilic here. And we have membrane proteins, and they are bringing molecules in and out, and we don't see any ATP being used. This diagram was an active transport. We would see ATP, but it's moving. You can see, well, I drew these in here. These white circles are only on this one side, so that's high concentration. Down here it's low concentration. So this would be a passive process because you have that differential. We can see it moving, and because it's being aided, this movement's being aided by a protein, that would fall under the category of facilitated diffusion. We're using a channel protein. So I don't know if I remember, these are, the whole thing is a phospholipid. That's what we call these little jellyfish looking things. Okay, remember, osmosis is only water moving from high to low concentration. Active transport goes the opposite. It goes, normally things go high to low. Active pushes things low to high. And it's pretty crazy. Plug my phone in. Sorry. Adboard needs to get on TikTok soon. All right, moving on. 16. There we go. What is the mechanism that generates that resting potential? So we have the, what's this question asking? Remember, what creates that resting potential is this sodium binding, inside the neuron to this. This is the sodium-potassium pump here. And once it's phosphorylated, then the sodium can leave. And then at the same time, potassium enters. So 3 sodiums out, 2 in. Remember, that's going to make the inside negative and the outside positive. So we are all negative when it comes to the charge of our neurons. All right. And also, we also reviewed this on that last paper exam. The fact that, being able to read this chart, this graph right here. Normally negative, acts potential, the nerve impulse comes, it shoots up positive just for a brief millisecond or 2, then pops back down to this negative threshold. Now, homologous chromosomes, very similar, but remember, they're not identical. Similar length, similar genes, similar shape, but not identical. Remember, one came from the mom, one came from the dad. So the answer is they're the same length in karyograms. That's the unit used to measure chromosomes. Where it doesn't pop up much. So that's a little tricky. Remember, they're not identical, different alleles. Remember, they came from different parents. Remember, prokaryotes don't have any chromosomes, right? And they can carry the same. They can, but they don't always carry the same allele. So that's IB, makes it tricky. Only in meiosis would be this random separation of homologous chromosomes. That's how you get all the variety that we see in living things that reproduce sexually, because you have that random separation, so you get randomly mixed up through this process of independent assortment. Like, remember this diagram here, say these are the paternal, these are the maternal, they just happen to end up on those 2 sides and they get separated. Well, what if that top one switched places, right? You have a different combination. All right. Which situation are alleles exchanged? Yes, crossing over in meiosis. When chiasmata are formed between non-sister chromatids. So these are sister chromatids from this chromosome. These are non-sister. This was went over all this in good detail in the very last videos that I posted a week and a half ago on topic 10. Remember, this always confused me, because when I think of chromosomes, I think of this, like an X. But you really should think of this. This is normally what chromosomes look like. And only look like this when they're going to be copied. So the chiasma is where they touch, also called a synapse. Okay? So that word might get thrown in there. So you want to be not too confused by that piece of vocabulary. So remember, a dominant allele can mask the effect of the recessive. Remember, if you have any dominant allele, you're gonna see it. It can't hide. Can't. But recessive alleles can hide and then pop back up later. That's what makes life interesting, right? Boom! Somebody in your family looks a little different, right? It's not the mailman or the milkman. I think I've heard in Spanish, Mexico, it's the milkman. In the United States, normal, whatever. Whenever I grew up, it was white culture. It's the mailman. If Anyway, let's see. Oh, codominant. That's interesting. Remember, that's when both show up. One doesn't totally dominate the other. They both show up together. There's the only thing I can think of is a type of horse that has red and white hair, so it looks like pink. But normally you have one get covered up by another. Let's move on. So, here we have a little cat question. Cat coat color. Okay, so there's a dominant capital M straight stripes, recessive M blotches and whorls, and then there's this A, the dominant produces 2 colors, but if it's the little a is all black, so there's no pattern, if you see that. So it asks you to do a cross of 2 double heterozygotes. What would be the outcome? So remember, heterozygous means they have one of each. So that's what we got here. This is one parent, that's another. Now to get these combinations, you just take this big one, match it up here, that's that. Then match it up here, that's this. Take the next one over. I'll go here, match up. There you go, little m, big A. I usually put it doesn't matter what the order is really. And then little m, little a. And then if you go through all the combinations, it's looking for the black offspring. So here you don't have to do all of them. You're just looking whichever ones have 2 little A's. So if you just kind of look through, no, big, they got a big. No, they got a big. Ooh, here we might get some. Not here. Oh, but here, see these 2 A's match up. Little a, little a. And then not here. Oh, little a, little a. It's going to be black. Gotta share this one with my wife because she loves cats. I love cats too. And I'm the one who introduced her to cats. I said, let's get a cat. They're great. She goes, I don't know. I'm like, oh, you try it. Man, it was over after that. Now we have 6 or 7. Okay. So, the key here to understanding how to do this, solve it, is, understanding what a heterozygous means, and then being able to do a little chart here. And again, on the test, they're not going to do something where you got to fill out the whole thing. So you just have to kind of know. Remember, normally you get a 9, 3, 3, 1 if you cross two heterozygotes. You get that 9, 3, 3, 1 ratio. All right, that would be your expected. Yes. 22. Okay. So, also in that last set of videos, talked a lot about Morgan's experiment. All right, he's the fruit fly person. So, what's the recombinant? Here are, remember here, gray body, vestigial wings. Vestigial means little stumpy things. And here is the genotype, and black body with a normal wings. Here we got that. So what we're looking for are 2 alleles that are different from anything we see in the parents. So BVG, we have that, but let's double check. We don't have any parent with a B, a plain B, and a VG plus. That doesn't happen. And we can see over here there must have been a crossing over. But like here, we have this one, we have that, we have this, we have that. So so these 2, nothing new, and there's nothing new here. That's just the same as the parent. But when I see this diagram, I kind of get freaked out. Oh, that's a lot of stuff. So just go slow. Remember, anything recombinant is going to be different from the parent. So what do I have here? Link, remember Morgan, all about linked genes. And offspring tend to look like the parents. You don't see the normal mixing of genes. Like, they most look like mom and dad, because those genes that give mom and dad their color, their wings, whatever it may be, are linked on the same chromosome. Mendel didn't know this because all of his stuff happened to be unlinked, on different chromosomes. So he just lucked out on that. Okay, so the characteristics of a species. Potential to interbreed fertile offspring. Yes, never fertile, able to reproduce. They form a population. Correct. The overproduction. That's part of natural selection. That usually is the case, but that's really a characteristic of natural selection, not definition of a species. Alright, let me go ahead and stop that video, and I'll finish hopefully with the next video.